JenniferADoudna, A Crack in Creation, Gene Editing and the Unthinkable Power to Control Evolution. Welcome to a world of genetic mastery, where evolution takes a back seat to the intentional control of creation. In A Crack in Creation, author JenniferADoudna unveils the groundbreaking CRISPR gene editing technology, its implications, and the questions it raises. As you delve into the world of DNA manipulation, explore its potential to cure genetic diseases, enhance agriculture, and even prevent conditions like HIV infection. However, with power comes responsibility. The ethical and regulatory issues surrounding this technology demand thorough deliberation. Witness the exhilarating reality of precise genetic therapy and its capacity to transform medicine while, at the same time, understand the ethical complexities involved as humanity ventures into shouldering the burden of controlling evolution.
Harnessing genetic code mastery. Throughout Earth's history, living organisms have evolved and diversified through a process characterized by random genetic variations. Nowadays, this traditional understanding of evolution is being challenged by new discoveries in genetic code manipulation. The author, a pioneer in this field, explores natural instances of gene editing, such as Kim's near-miraculous recovery from WHIM syndrome. As scientists continue to uncover the mysteries of genetics, there is increasing potential for intentionally modifying the genetic code to cure diseases and improve lives. For eons, the Earth's living organisms have undergone a fascinating transformation, driven by chance variations in their genetic makeup. This captivating array of biological diversity has been traditionally explained by Darwinian evolution. However, contemporary research now offers fresh perspectives on this age-old concept. A groundbreaking era of biological mastery is emerging, and the author is at the forefront of this scientific revolution. By exploring the targeted modification of genetic codes, we enter a realm where natural evolution is no longer the sole determinant of life's changing course. Contrary to what one may initially think, tweaking the genetic code isn't necessarily unnatural. In fact, nature has its own ways of conducting gene editing. A case in point is Kim, a patient who baffled National Institutes of Health, NIH, scientists in 2013. Despite her diagnosis with WHIM syndrome, a debilitating immunodeficiency disease caused by a single DNA spelling error, Kim was, against all odds, symptom-free. Upon closer examination, experts found that an astounding 35 million letters were missing from one of her chromosomes, and her remaining DNA was in disarray. The most plausible explanation was chromothripsis, a phenomenon in which a chromosome unexpectedly shatters and rearranges its genes. This tumultuous process seemingly eliminated Kim's genetic error, resulting in her miraculous recovery. In essence, nature had conducted its own edit on Kim's genome, significantly improving her well-being. This raises a compelling question: what if we could go beyond relying on serendipity, and use our advancing knowledge to purposefully correct genetic errors, thereby conquering disorders and enhancing lives? Delve further into this captivating domain of research, as the next section uncovers more insights into the world of genetic code mastery. Decoding gene editing evolution. The genome, comprising all our genetic information, encodes our physical attributes and susceptibility to diseases. Genes, made of DNA molecules, adenine, guanine, cytosine, and thymine, are responsible for specific bodily functions. The gene editing quest began with the observation that viruses could insert their DNA into cells. The first breakthrough occurred in the 1980s using homologous recombination, but it lacked therapeutic viability. Recurrently disappointing attempts paved the way for the discovery of CRISPR, simplifying and revolutionizing the gene editing process. Diving into the world of gene editing, it's crucial to understand the lingo. The genome is the sum of our genetic information within our cells, governing our physical traits and even disease susceptibility. It consists of DNA molecules, adenine, A, guanine, G, cytosine, C, and thymine, T, which, in essence, are the genetic alphabet. Human DNA is packaged into chromosomes, which house groups called genes, responsible for specific biological functions. Keeping this in mind, let's explore the journey of gene editing, a tale that started with the observation that viruses can insert their DNA into cells and even join it with bacterial chromosomes. Triggered by this observation, the first key gene editing discovery occurred in the 1980s, when Mario Capecchi and Oliver Smithies found a way to swap defective genes with healthy ones using homologous recombination. However, the technique proved therapeutically unreliable, as it succeeded in a mere 1% of attempts. Throughout the 1990s and 2000s, scientists explored alternative techniques, but all fell short due to their complexity and unfeasible clinical applications. However, a turning point arrived when researchers identified a DNA region in bacteria described as CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. Simply put, the DNA repeated itself precisely over specific intervals. This groundbreaking discovery paved the way for a simpler and more effective gene editing technique that could revolutionize medicine and genetics. CRISPR has emerged as a promising tool, ushering in a new era of genetic potential. Unraveling CRISPR's secrets. CRISPR, a gene editing tool, features repeating DNA sequences found in bacterial genomes, holding the key to a natural defense mechanism against viruses. Central to bacterial immunity, CRISPR's function as molecular vaccination cards, storing traces of previous viral encounters. To disable viruses, CRISPRs utilize three main components: Cas9, a protein coded by associated genes, CRISPR RNA as a guiding messenger, and tracrRNA as an activation assistant. This wonder of nature prompted scientists to ask if CRISPRs could be modified to target and splice other types of DNA. CRISPR, the groundbreaking gene editing mechanism, is a region of bacterial DNA characterized by repeated genetic sequences. Found interspersed amongst these repetitions are similarly sized chunks called spacer sequences. The prevalence of CRISPRs in bacterial DNA signaled a vital purpose, sparking scientific curiosity for better understanding. In the mid-2000s, researchers pinpointed the function. They discovered that the spacer sequences closely matched the DNA of bacterial viruses. Unveiling the connection, they realized CRISPRs functioned as an essential component of the bacterial immune system, fending off viruses. Like molecular vaccination cards, bacteria use CRISPRs to store memories of past viral encounters in spacer sequences, granting them the ability to recognize and annihilate those viruses during subsequent infections. This remarkable defense relies on three key components. First, Cas9, a special protein coded by CRISPR-associated genes, acts as the primary player in slicing through and deactivating the invader's DNA. Second, CRISPR RNA, a messenger molecule within cells, directs the Cas9 protein towards its cutting destination. Lastly, tracrRNA aids in activating this DNA cutting process. Having decoded these natural phenomena, scientists ventured one step further with a burning question: could the CRISPR's virus-slaying capability be harnessed to target and splice other forms of DNA? This inquiry opened the door to a world of potential applications in genetics, agriculture, and medicine. The power of CRISPR gene editing. CRISPR stands for a revolutionary gene editing tool that allows scientists to precisely match, cut, and replace specific DNA sequences. By utilizing associated CRISPR RNA, the cutting protein Cas9 can locate exact matches in foreign DNA and remove them. During the natural repair phase, scientists can swiftly insert alternative DNA sections, offering endless possibilities for genetic research and transforming the landscape of medicine. Let's dive deeper into the world of CRISPR and its extraordinary capabilities. CRISPR, clustered regularly interspaced short palindromic repeats, is essentially a molecular cutting machine with a keen sense of accuracy. What sets this method apart is the unmatched precision in identifying and removing specific DNA sequences. First, CRISPR RNA recognizes and guides the Cas9 protein to a target location in foreign DNA, where there's an exact match with the original CRISPR DNA spacer sequence. Once the Cas9 protein has served its purpose, it's led away by the guide RNA, leaving behind the altered DNA. During the natural repair process, scientists get a brief opportunity to introduce a new DNA section in the cut location. This groundbreaking technology enables unprecedented control over gene editing. In 2012, a monumental paper co-authored by Emmanuel Charpentier and the author showcased CRISPR's potential in manipulating jellyfish DNA. Being cheap and user-friendly, the CRISPR method immediately drew attention and inspired further research. In 2013, Kiran Musunuru from Harvard used CRISPR to correct a gene mutation responsible for sickle cell anemia, proving its potential to change the face of medicine. CRISPR empowers scientists with a higher degree of control over genetic information, solidifying its position as the most valuable tool in the world of genetic research. As we continue to unlock CRISPR's potential, it can potentially transform medicine and our understanding of genetics. CRISPR's agricultural revolution. Imagine a world where our food gets healthier, more resilient, and sustainable, all thanks to the power of gene editing. CRISPR technology carries great potential to transform the world of agriculture, from rescuing the citrus industry from the devastating Huanglongbing disease to reducing unhealthy trans fats in soybean oil. Meat industry also stands to benefit, with innovations like the EnviroPig that impacts the environment less, and the potential to create cows without horns, improving animal welfare. The future of food production may be revolutionized by these groundbreaking applications of gene editing. The rise of CRISPR gene editing technology has unleashed numerous possibilities for innovations, even including the potential creation of woolly mammoths or unicorns. More importantly, though, are the practical implications of CRISPR in agriculture, where this revolutionary method can enable higher yields, stronger crops and healthier food. Citrus trees plagued by Huanglongbing, or yellow dragon disease, could be saved by CRISPR, with the orchards of Florida and California under imminent threat. Another impactful application of CRISPR is in soybean oil, a widely consumed staple with unhealthy levels of trans fats. Gene editing could alter the soybean genetics to reduce these fatty acids, providing healthier oil for millions of consumers. Beyond plants, livestock also stands to gain from CRISPR technology. Canadian researchers have developed an EnviroPig, a genetically modified pig containing a gene from E. coli bacteria that improves digestion, cutting the phosphorus content in its manure by an impressive 75%. This breakthrough reduces the harmful effects of phosphorus-laden manure, which can generate destructive algal blooms when entering water systems. Furthermore, CRISPR could improve animal welfare in the livestock industry. For instance, cows are typically subjected to painful and stressful procedures to remove their horns. By utilizing gene editing, it could be possible to create cows without horns altogether, sparing them unnecessary suffering. The immense potential of CRISPR technology ushers in a new era for agriculture, promising healthier, more resilient, and sustainable food for generations to come. CRISPR, unlocking genetic cures. Over 7,000 human genetic diseases stem from individual gene mutations, but CRISPR technology brings hope for potential cures. Gene editing could revolutionize medicine by paving the way for precise genetic therapies. CRISPR has shown promise in HIV resistance, Duchenne muscular dystrophy, and cancer research. Despite its potential, gene editing also harbors risks as we navigate being in control of our evolution. Imagine a world where countless medical mysteries are suddenly within reach of being solved. Welcome to CRISPR, a genetic editing tool transforming the way we approach science and medicine. While we face over 7,000 genetic diseases caused by single gene mutations, CRISPR offers hope for groundbreaking cures. Take HIV for instance, millions worldwide suffer from this devastating virus. However, a small group of individuals possess a remarkable natural resistance. The key lies in the CCR5 gene mutation coding for a specific protein. Current research strongly suggests that CRISPR could edit the CCR5 gene in non-resistant subjects, opening the door to HIV prevention. Similarly, Duchenne muscular dystrophy, DMD, proves a life-altering and fatal genetic disorder, affecting 1 in 3,600 male infants. As this cruel disease leads to severe muscular degeneration, often confining children to wheelchairs by age 10, CRISPR offers a glimmer of hope. Recent studies on mice highlight the potential of CRISPR in finding a cure for DMD, a disease caused by the mutation of the DMD gene. The fight against cancer could also experience a significant boost thanks to CRISPR. Cancer arises from DNA mutations, which can be inherited or acquired through lifestyle choices like smoking. CRISPR technology promises an opportunity to eliminate such mutations, potentially resulting in innovative treatments or even outright cancer prevention. While the benefits of gene editing are abundant, it's crucial to remember that this power over our genes and evolution comes with risks. As we unlock the secrets of CRISPR, we must also carefully examine and weigh the ethical implications of editing the very fabric of life. CRISPR, ethical implications. The excitement surrounding CRISPR's potential in 2014 led to questionable propositions, such as creating CRISPR babies. This raised ethical concerns about gene editing becoming too easy and accessible, as well as the possibility of creating designer babies. Recognizing these concerns, the author and other experts published a white paper in 2015 focused on ethical implications of gene editing in the human germline. They called for a halt to such research until further deliberations could take place. Ultimately, society must decide how to use CRISPR, and education on the matter is crucial. Just 2 years after a groundbreaking discovery in gene editing, the skyrocketing interest in CRISPR had people captivated. Samuel Sternberg, a then PhD student who worked with the author, received a startling offer, joining a startup that sought to create the first CRISPR baby. He declined, but it brought forth pressing questions: was gene editing becoming dangerously easy? What were the ethical concerns surrounding this rapidly advancing technology? CRISPR undoubtedly held the potential to do incredible good by preventing genetic diseases. However, it also opened the doors to designer babies, those whose genes had been manipulated to possess specific traits or characteristics. This raised numerous ethical dilemmas, such as selecting a baby's gender or enhancing them with desirable traits like increased muscle mass. The author even worried about the potential for the abuse of CRISPR, going as far as dreaming about Adolf Hitler using her discovery for devious ends. In an attempt to spur a larger conversation about gene editing, the author and other experts published a white paper in 2015 discussing its ethical implications. They delved into the topic of human germline editing, referring to cells transmitting genetic information during reproduction. The authors urged the scientific community to hold off on further germline research until a wider discussion tackling social, ethical, and philosophical conundrums could occur. In conclusion, the power of CRISPR must be entrusted to society as a whole. To make informed decisions, it is imperative people first become educated on the matter, enabling a productive dialogue on the future and ethical considerations of gene editing. Navigating gene editing dilemmas. As gene editing advances, 3 key themes must be carefully considered: safety, ethics, and regulation. The author envisions a future where germline editing is safe enough for clinical use, offering potential solutions for disease-causing mutations. However, society faces a dangerous ethical line between improving health and enhancing attributes like intelligence or athleticism, which could lead to a divide favoring the wealthy. Establishing comprehensive international regulations and fostering diverse conversations are essential to responsibly guide this groundbreaking technology. Before embracing gene editing, society must examine 3 core principles: safety, ethics, and regulation. The author suggests that, eventually, germline editing will reach a level of safety suitable for clinical use. With millions of natural genetic mutations occurring within the human body each second, the potential benefits of eliminating disease-causing genes using CRISPR may outweigh the risks of unintended mutations. Despite safety advancements, ethical concerns arise when distinguishing between improving health and enhancing traits such as intelligence, athleticism, or beauty. This blurry line raises questions about genetics becoming a resource primarily for the wealthy, who could disproportionately benefit from germline editing advancements. Rather than prohibiting germline editing entirely, it is crucial to understand and address legitimate objections in order to harness this technology responsibly. Lastly, they highlight the importance of regulations to manage human germline modifications. Governments should actively participate in overseeing this area of research, and ideally, a global consensus on policy would be established. Encouragingly, progress is already being made with events like the 2015 International Summit on Human Gene Editing, which brought together politicians and scientists for crucial discussions regarding this technology. In conclusion, as gene editing continues to progress, navigating the complex issues of safety, ethics, and regulation is essential. By fostering open dialogue and working towards global consensus, society can responsibly guide the development and implementation of this groundbreaking technology in the best interest of all.
In essence, ACrackinCreation presents CRISPR gene editing technology and its potential to revolutionize medicine, agriculture, and our understanding of genetics. Its potential seems so limitless. The book provokes questions. Are we playing God? How will this newfound power affect society at large? Doudna acknowledges the ethical minefield and stresses the necessity for open discussions among the scientific community, policymakers, and the public to examine our responsibilities and establish a regulatory framework in the interest of all. The confluence of safety, ethics, and regulation is central to the book's message, emphasizing the importance of engaging in such conversations before advancing further into this fascinating realm of genetic manipulation.